Nice, thank you. You'll have to humor me. I feel compelled to tell the same joke every year, which is I'd like to thank the organizers. I will not abuse my privilege of being able to control the agenda and speak for 45 minutes, but thank you for considering that I might be able to do that. All right, so Lineage Cell Therapeutics, we are not a professional conference organizing company. This is a side hustle. But what we do is we are trying to be pioneers of an emerging technology, which is the differentiation of specific cell types and then the transplant of those cell types into people in order to recover function or ability. As we are a publicly traded company, I have to caution you about not only the opportunities but also the risks of investing. And if you need to know more about such topics, please review our public filings with the SEC. All right, we are a local company. We're right up the road in Carlsbad. We have a small R&D facility there, and all of our clinical material and all of our process development and everything we do in manufacturing is conducted outside of Jerusalem, a suburb of Jerusalem, with a large team that we have there. We have about 75 employees overall worldwide. Our view, therapeutically, is that in certain settings, cells can do more than small molecules or antibodies. And if you think about it this way, small molecules are very targeted. They usually hit a single pathway, which can be very powerful. Aspirin's a pretty good therapeutic for a headache. But in degenerative diseases and certain traumatic conditions, what's happened to the cell is it's completely destroyed or it's completely dysfunctional. So there's a lot more than one pathway that's going on. So our view is those solutions might require much more powerful interventions. How we endeavor to do this is we manufacture basically replacement parts. So we have cell lines that are pluripotent, which means they have the ability to become any of the cell types of the human body: brain, bone, lung, kidney, heart, everything that you are, all the 200 plus cell types that comprise your body, they all start from an undifferentiated, call it blank slate cell. So we utilize those cell lines and the power of those cells to become anything. But we're actually even more excited by the fact that those cells are self-renewing, which means we have an unlimited supply of them, right? You give the cells sugar water, they keep growing and dividing, and you basically have an unlimited amount of that. And then we give those cells instructions to become whatever cell type it is we are interested in. This doesn't involve any gene editing or anything like that. We are just using natural developmental biological signals which turn cells from something blank into something specific and functional: kidney, heart, etc. In this case, oligodendrocyte progenitor is kind of a nice choice because that's a primary cell of the spinal cord. We have a whole bunch of therapeutic efforts that are all built on this same approach of manufacturing specific cell types. The most advanced one is actually in the setting of dry age-related macular degeneration, where we make RPE cells. And that approach led to a nearly $700 million deal with our partners Roche and Genentech. What we like about that is it helps to validate other things that we're doing, right? A big pharma company came in, gave us a $50 million upfront payment and sort of validated this successful approach in the eye. And we're looking to recreate or reproduce those effects in other settings where we think a cell could drive a better outcome than a small molecule or an antibody or some other interventions. So I will only be talking about oligodendrocyte progenitor cell transplants today. There have been 30 people who have been administered this therapy. One of them is here today. The idea is pretty straightforward, right? If you have a traumatic injury and these oligodendrocytes are destroyed, either through the original injury or through the inflammatory cascade, which occurs thereafter, if you don't have that substrate, you don't have that material, it's really hard to engineer that response and restructure that response. And so the cells that we manufacture, the oligodendrocyte progenitor cells, they are the myelin-creating cells of the spinal cord, and that's, of course, the material that's carrying that signal and transducing that signal between the brain and the extremities. We've been doing some interesting things on the manufacturing side. A lot of people think about cell therapy as being a very personalized or what's called an autologous therapy, meaning I generate the cells for my own medicine, right? An N of 1, right? Somehow cells are taken from my body, manipulated in some way, and then put back in, and that's my, quote, medicine. What we have is an off-the-shelf solution, meaning we'll grow millions and billions of cells, and those are the same cells. They are able to be put into any patient. They're not rejected. People go on to very short-term immunosuppression, just 60 days, and they're durable. They remain inside the patient, and nothing weird is growing. You don't have people growing ears where their spinal cords belong and things like that, right? A lot of that is stemming from early forays into cell therapy that some of which were very unsuccessful and a bit scary, but it's 2024, and the technology to control the material you're making and understand what you're making has advanced not just at Lineage, but a lot of places. And so the field has really become positioned for a lot of success by overcoming some of the early hurdles. We haven't seen any of those, really scary things that you might have read about years ago. We haven't seen anything like that in the context of any of our clinical efforts to date. If you're able to put oligodendrocyte progenitor cells into someone, there are a few things that we think are good, which could be happening. They can be remyelinating, they can be preventing the cavitation, they can be secreting positive neurotrophic factors. Again, the idea is quite straightforward and easy to understand, i.e. replacement parts. We have been following. It's very important that if you put the cells in, they need to be durable, right? They're gonna engraft. They're gonna become part of your body forever and ever and ever. And this is one of the longest-running studies of its kind. So we have patient data going out 5 and in some cases even 10 years or longer. And so that's a tremendous body of safety data for this intervention, which is really encouraging because there are few therapies that have that kind of long-term follow-up. So that's a very important aspect of what we're doing. The motor recovery that we saw, and keeping in mind, of course, we've talked a lot about variability, and it's very difficult to find a truly matched historical control. You find yourself relying on the input from the surgeons and the other caregivers to really understand what did you think might have happened with this person. But our best efforts, working with experts outside of the field to try to find a matched control, suggest to us that there's a signal that we have enjoyed here in the phase 1 study. Most notably, almost everyone on the study, this is a group of 25 cervical patients, the first 5 were thoracic, but of the 25 cervical patients, almost everyone improved somewhat. So that's very encouraging. But of course, where we need to go with this therapy is to do some form of a controlled study and really answer the question, how much of that benefit is attributable to the cells that we have transplanted? And by the way, this is a single transplant, or a one-time transplant, I should say. If we can improve performance, if we can increase the quality of life, if we can reduce the reliance on different providers, obviously there's a wonderful economic and social benefit from doing so. And it's actually a point of great frustration for me that when we talk with people who are experiencing spinal cord injuries and say things like, as Jared was saying earlier, any improvement, like let me be the arbiter of what matters. Like, let me have the improvement. I'll figure out what to do with it. It's really powerful because I think expectations are really misapplied. And frankly, just sidebar, part of this conference is to get alignment and understanding throughout the field that maybe our expectations are too high. We need a place to start. We need a success. We need a win in this field. Maybe it'd be Lineage, maybe it'd be someone else, maybe it'd be a bunch of us. That would be great. But we really need to start to reset expectations on what matters. All right, off the soapbox and back to the other soapbox. If you're going to be putting something into someone and it's going to stick around for their entire life, you really want it to be safe and well tolerated. This is really compelling data from my perspective because when I think about new therapeutics, of course, I'm always thinking about FDA as the gatekeeper. They're the ones who say, yes, this can reach the patient or no. And they do that by assessing the risks and the rewards. So this is quite interesting data from that 25-patient study that was previously performed and completed. 534 adverse events. Only one was potentially associated with our cells. It was a grade 2 dysesthesia. It auto-resolved. That's a really good safety profile. And it's another thing that's kind of interesting and broadly applicable, we believe, to cell transplants. They're really well tolerated. Unlike some molecules that, in different conditions cause all sorts of adverse events, cell therapy has been extremely well tolerated. So when you think about the future opportunity of a product being approved and you think about running that through the filter of risk and benefit, it's really nice that our adverse event profiles and the tolerance of these therapies has been extremely good. Fairly recent publications here from both of these studies, the thoracic and cervical studies. Again, 30 patients in total have been recorded and tracked for very long periods of time, which was encouraging. So we've got the phase 1, 2A data. It looks like the signal is very well tolerated and safe. But, here's the big but, not everything that you test, even in the setting of in a human setting, is necessarily ready for prime time. Our philosophy, if you will, with respect to cell therapy, is that you're really not in the game if you don't meet these criteria, right? It's not enough to just have an interesting phase 1 signal. You have to have control of your process. You have to be able to scale. It has to be able to be reproducible. Not every cell therapy has this. They might have some interesting even clinical data, but not everyone really has what it takes to go all the way. And since this is an investor forum and you think about, look, how do we attract capital to be able to do all of the things that we want to do, you have to tick all of these boxes. You can't fake it till you make it in this industry. You really have to have something which is going to be capable of being commercially successful through the entire channel and the entire ecosystem. So these are things we think greatly about, and we saw some deficiencies in this program. One of them was we saw an opportunity to improve the quality of the material, the purity of the material we manufacture. So this is just a comparison, these gray bars, and it's a measurement, and I'm just shorthanding, but it's a measurement of impurities. And so you can see in a couple of cases impurities into the high teens, even 20%. These are cells in the clinical material that maybe aren't what we were really trying to make. That's the stuff that went into people, right? That's the stuff that drove those clinical outcomes. The orange bars, that's what we're making now. So what we did was we deconstructed the process, reevaluated it, changed how the cells were being manufactured in order to come up with a much more pure population, really trying to drive the differentiation and the lineage of the production further and further toward pure material and toward the oligodendrocyte progenitors. We more recently brought into effect, and it is a technology that didn't exist when these patients were being treated, but the bioinformatics that we're looking at. And one kind of cool example here is, if this is a laser, it's not. Anyway, top left, purple, epithelial cells. So they're markers. Now, we don't know that they're really epithelial cells, but markers of epithelial identity in clinical material probably aren't going to do anything good. Might not do anything bad, but probably aren't helping. So when we broke down the process and reconstructed it, we were able to eliminate that undesirable cell population. This, again, is something that's happening not just here in our hands. There is a revolution happening in cell transplants where the industry's ability to control the stuff we're making is just going absolutely off the rails, and it's really, really cool stuff. So being able to see through single-cell RNA-seq data like this exactly what you're making and following the kinetics of what you're making over time to really understand it has elevated to the point where now FDA expects it. So that's a great thing for the field and our understanding and knowledge of what we're doing when we make these cells. We also see an opportunity to improve how the cells are delivered, right? This is all external to the patient now. The original way of delivering the cells involved this big scaffold. There were a lot of parts. You had to bolt it to the bed. You had to autoclave and sterilize all the pieces. It made the surgical field really big. It was cumbersome. Dr. Fessler knows. He worked with this. And one of the really annoying features of that old system was that you had to disconnect the patient from respiration while you were administering the cells. So, you're working in the spinal cord, you're delivering cells, and by the way, the patient's not breathing. So you kind of need to go quickly. I just realized there's a joke there about we used a site that's called Rush. So, okay. I don't know why that never occurred to me before. So anyway, we thought it would be really cool if you could miniaturize this. And we entered into an alliance with a device company. I think the CEO of that company is here today. The new device still holds the needle. It's the same kind of needle. The flow path is still very straightforward. But the device is now attached to the patient. So there's retractors. It's actually connected to the patient. So now visualize this. Patient's on the table, respiration, chest cavity going up and down, device holding the needle up and down. You can take 4, 5, 6 minutes to deliver the cells to the cord now. So you don't have that same urgency, you don't have that same race. We also recreated the formulation for the cells. So it used to be that you had hours, I think it was about 4 hours of taking the cells, washing them, plating them, preparing them, counting them, checking them, blah, 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 blah. Now, you're taking them from a frozen state and going into the patient just 5 minutes after thawing. So we've eliminated a lot of the commercial headaches and the usability annoyances of this material by improving the control of how we make it, the scale of how we make it, which is also up 10 to 20x, as well as how we're delivering it externally. So we're trying to be thoughtful about all of the different components that go into a successful therapy. The next thing that we're going to do is to run a study of that new device. So that new device hasn't been used yet with our cells, so we're aiming to initiate a 6 to 10 patient study. It will be ostensibly to demonstrate that that new device is suitable and would be utilized in a much larger study. But it also affords us the opportunity to be able to do a set of chronic patients. We've never put our cells into chronic patients before, chronic injury patients before. So this is the first time we're going to see if we identify any sort of functional benefit or changes for someone who has an older injury and getting these cells. It's a really important question, and we just have this wonderful opportunity to piggyback on this otherwise very straightforward safety study and access into a new patient population in that way. So very excited about that. So this, as I say, is one of the longest running studies. It's been around, so many learnings. I was not involved in this at the beginning. We're sort of standing on the shoulders of some giants here, but this is now a study that there have been tremendous learnings that have come out of it. I have seen our job, our role to build upon that, to improve it, to set it up for success before going into the next study. And so we're going to make sure that we have those characteristics in place. And that is a nice feature for us, is that it gives us time to work on all the planning and preparation that you're hearing about today, all the scary stuff that you hear today about how hard this is and variability and all of that. We have now this conference and these kinds of conversations to work on that so that when we initiate a larger controlled study to answer the question, how much benefit do you get from cells, we'll be set up for as much possible success and sensitivity as we can be. So OPC1, this is straightforward this is a straightforward cell transplant program. It has a lot of regulatory advantages. It has been the proud recipient of a significant amount of CIRM funding, so we're really happy. Thank you again, CIRM, for also supporting this conference. You know, I just couldn't be prouder about all the people who have been involved in this program and all the good work that we're trying to continue. So thank you very much, and look forward to the rest of the conference.